we have a lot of interesting stuff. We'll start then.
Okay.
So this tradition of hearing about the Science Nobel Prizes goes back many years. I think it started with a discussion with Ellen Tartikoff from the medical school, who is with us today, about how we could make recognition of the Science Nobel Prizes an interdisciplinary event. And over the years, we've also had co-sponsorship from the chemistry and the biology departments. But so today we're going to hear from my physics colleague, Kurt Hinterbichler, on the Nobel Prize in Physics, from Ron Conlon of the Department of Genetics and Genome Sciences on the Chemistry Prize, and from Donald Anthony of the Department of Medicine on the prize in Physiology or Medicine. Because our time is limited, we're asking each speaker to keep to a maximum of 15 minutes so as to allow time for questions. And we'll save our questions until all three speakers have given their segments. But do feel free to enter any questions you have in the chat box. So we start with the 2020 Nobel Prize in Physics, which was given for theoretical and experimental work in cosmology. And we hear from Professor Kurt Hinterbichler, who is himself an expert in this area. Over to you, Kurt.
Okay, thank you. You can see the slides okay?
Yeah, it's good.
Okay, so the 2020 Nobel Prize in Physics was awarded to the physics of black holes, both on the mathematical theoretical side and on the observational side. So half of the prize was to Roger Penrose for the discovery that black hole formation is a robust prediction of the general theory of relativity. And the other half was split between Reinhard Genzel and Andrea Ghez for the discovery of a supermassive compact object at the center of our galaxy. So the idea of a black hole has been around for a while, since the late 18th century. And it starts by imagining the escape velocity. So if I sit on the surface of the Earth and I throw a baseball upwards, it comes down. But if you throw it fast enough, it will have enough energy to escape the pull of gravity and go off to infinity. And the velocity with which that happens is this escape velocity v, which is given by this formula here, where g is the Newton's gravitational constant, m is the mass of the object you're sitting on, such as the Earth, and r is the radius of the object you're sitting at, such as the Earth. So as you make the object more and more massive, or you make it smaller and smaller, this escape velocity goes higher and higher. So for the Earth, it's about 11 kilometers a second, 25,000 miles an hour. For the Sun, it's 600 kilometers per second. But you could imagine that maybe there's an object that is so massive and so compact that the escape velocity actually starts to approach and maybe exceed the speed of light, which is 300,000 kilometers per second. So if you had an object which even light couldn't escape from, we wouldn't be able to see it. It would be completely dark. And this idea was come upon independently by two different polymaths in the late 18th century. So Laplace in France wrote that the largest bodies in the universe may well be invisible by reason of their magnitude, because even the light couldn't escape from it. And even earlier than that, John Michell in England went further and said, if there are such objects, if there are other luminous bodies which would happen to revolve around them, we might be able to detect these objects by looking at the luminous objects that are revolving around them. And this is precisely, we'll see, what the Nobel Prize was awarded for achieving. Okay, so we're talking about objects that are very massive and where speeds are approaching the speed of light. The proper framework for this is general relativity, which was developed by 1915 by Albert Einstein. So here are the field equations of general relativity. So in this theory, gravity is described as an effect of curved spacetime, and it extends Newton's theory to make it compatible with special relativity, so you can actually start to ask questions about what's happening as speeds approach the speed of light, and gravity is important. So no sooner had this theory been developed than a solution was found, which is called the Schwarzschild solution. In the same year that it was developed, 1915, Karl Schwarzschild, while fighting on the Eastern Front of World War I, managed to find time to derive an exact solution to Einstein's fields equation. And the solution has the feature of, it's a spherically symmetric solution. So this solution is what we now call a black hole. there's a singularity at the center. So all the mass of the object is concentrated at the center. And then surrounding it at this radius here is the event horizon, which is like a point of no return beyond which light can't escape from. So Einstein was very impressed by this derivation. In a letter from Einstein to Schwarzschild, he mentioned that he hadn't expected that a solution like this was even possible. He thought the equations were too complicated to ever admit an exact solution. And then, as you see from his date of death, Schwarzschild died in the war just shortly after. So we have these solutions within the theory. The question now is, are these solutions actually recognized or actually realized in the real world, in nature? Is it possible for matter that we know about to evolve towards solutions like this? So this leads one to consider gravitational collapse. So if you think about a star is a giant nuclear reaction, a nuclear fusion reaction that's held up against the pressure, against the downward pull of gravity by the pressure of this nuclear reaction. And when the nuclear fuel runs out, it starts to collapse. And for masses that are small enough, the end state of this collapse is a so-called white dwarf star, which is held up by electron degeneracy pressure. So the pressure that results from electrons, the Pauli exclusion principle, electrons don't like to be in the same place at the same time, and this leads to a kind of pressure which is sufficient to stop the gravitational mass so long as the mass of the object is not greater than about 1.4 solar masses. So these white dwarfs are objects which are very, they're about as massive as the sun, but they're as small as the Earth. So very, very dense. If the masses are larger than that, so greater than about 1.4, but less than about 3 or 4 solar masses, then this electron degeneracy pressure is insufficient to stop further collapse, and it collapses further. The electrons combine into the protons and all form neutrons, and you get an end state which is a big giant chunk of neutrons, essentially, a neutron star, which is held up by degeneracy pressures of the neutrons. And this object is even more dense. It's only about the size of a city, but still as massive as a star. Beyond this limit, so beyond 3 or 4 solar masses, there's no known force or mechanism that can prevent a further collapse. And the stars in the real world that we see out there have masses much larger than this. Some stars have masses up to 100 solar masses and possibly more. So there are definitely stars out there which there's no known way for them to stop collapsing once they ran out of fuel. And these very massive stars also happen to have very short lifetimes. So there should be objects out there that result from this collapse process. So mathematically, another landmark paper was this paper by Oppenheimer and Snyder, the same Oppenheimer that led the Manhattan Project to develop the atom bomb during World War II, where they showed, using the equations of general relativity, that if I have some collection of matter and I let it collapse, it will in fact form this Schwarzschild solution, this black hole solution. So then the question becomes, all of these mathematical investigations and the Schwarzschild solution and so on up to this point invoked spherical symmetry. So there are solutions which are exactly spherically symmetric. So you can imagine that this singularity at the center is somehow an artifact of spherical symmetry. So just like the 1/r Coulomb force has a divergence when r goes to 0, but in reality, everything's kind of smeared out. You can imagine the same thing here, that if you have a gravitational collapse where everything's not exactly spherically symmetric, that maybe as the things collapse, it just kind of splatters and you don't ever get this actual singular solution. So that was a question, and the first part of the Nobel Prize was essentially an answer to that question. And the answer is this singular solution is not an artifact of spherical symmetry, that it's in fact a generic property of the gravitational collapse. So what Penrose proved, starting with this paper in 1964, is that under quite general conditions, even without spherical symmetry, that you will get this gravitational collapse, and the theory of general relativity predicts that you get these black hole-like solutions with singularities. And this paper is rather remarkable. It's only 2.5 pages long. Most of that is introduction and conclusion. The actual proof is only about half a page or so. Okay, so the second half of the prize was awarded for actually observing black holes in the real world. So one way to do this is by looking for things that you can see, like stars that are orbiting around seemingly nothing. So there's a region in the sky known as Sagittarius A star, which is a subregion of Sagittarius A, which is a point in the sky that's towards the center of our galaxy. So here is the constellation Sagittarius, the teapot, and then this X here is the direction towards our galactic center. And this region has been known since the early 1930s as a radio source in the sky. And this region, Sagittarius A star, is a very bright, compact, very bright in radio and compact subcomponent of this Sagittarius A region, which was found in 1974. And it's called A star because in chemistry, the star denotes an excited state. And this was a very exciting region of the sky. So the second half of the Nobel Prize, what Genzel and Ghez did, they each independently led an observational group which studied and observed this central region of the galaxy for a period of over 20 years and accumulated strong evidence for the existence of a black hole that's in the center of our galaxy, a very, very massive black hole. And they did this by essentially observing the motion of stars around the black hole. So here is a movie. And this is not a simulation. This is actual data, actual images taken over a period of 20 years or so and made into a movie. And what you see are these stars orbiting around. So this star in the middle, particularly, you can see it's on an elliptical orbit, and it's orbiting something that's completely invisible. And if you draw the picture of the orbits, this is kind of what they look like of the various stars. So the scale here, all of this fits into about a square arc second of the sky. So the resolution you need to see this is very, very fine. The kind of resolution you need to see these stars is equivalent to distinguishing between the left and right eye of a person that's standing about 250 kilometers away. So by studying these orbits and using just ordinary Newtonian dynamics, you can figure out what the mass of this central object is, and it's about 4 million solar masses. So it's a giant, very, very massive object, far more than any known star. And furthermore, you can put an upper limit on how big it is. So some of these stars, like S2 here and S14, they get very, very close to the object and they swing around it. So the fact that they're not destroyed by this object means that the object has to be smaller than this closest approach. And the scale here, if you look in the bottom right corner here, the very small dot on the top there is our solar system. So the closest approach of these stars is about the size of our solar system. So whatever this object is, it has to be smaller than our solar system, but it has to be 4 million times more massive than the sun. And the only such object that we know about is a giant black hole with a Schwarzschild, which would have a Schwarzschild radius of about 14 times the radius of the sun. So the difficulties they had to overcome to observe this are considerable, mostly having to do with the very fine angular resolution that you need to see these stars. So you need very large telescopes. They used some of the largest telescopes in the world. Here's one of them, the Very Large Telescope Array in Chile. And you need advanced interferometers to be able to combine the light of multiple telescopes to get even better resolution. And you need to worry about the atmosphere because there's atmospheric turbulence that can ruin the resolution. So you need to take very short exposure times and stack them and use adaptive optics and all kinds of things to be able to achieve this kind of resolution. So this observation of the black hole, these supermassive black holes, it's still a mystery how they form, because obviously there's no stars that are that big to collapse to form a black hole. So they must have maybe combined from a bunch of little black holes or somehow been there primordially at the beginning of the universe. That's still an open question. And we now think that there's a giant massive black hole like this at the center of every galaxy. So on the bottom here, you see an image, the first direct image of a black hole that was taken of a very, very massive black hole, billions of solar masses, that's present in the galaxy M87, about 50 million light years away. This is the first ever direct image of a black hole. And they're also working on imaging the one that's in the center of our galaxy. We've also seen smaller black holes, these stellar mass black holes, both from seeing things orbit and from gravitational waves now, seeing the gravitational waves of when they collide. But there's still a mystery about what about intermediate black holes that are, say, 10,000 times the mass of the sun, but not 1,000,000 times the mass of the sun. Those, we still don't know whether those exist. And there's all sorts of open questions about how whether they exist, how they form, how these supermassive black holes form, and so on. But the existence of black holes is now a more or less settled question. Yeah, I'll stop there.
Well, thank you very much, Kurt. That's very interesting. And you managed to get a lot of information into that brief time. So thank you very much for being so careful about the timing there.
Yep.
So now we go on to the second prize, which was the 2020 Nobel Prize in Chemistry. And that was awarded for some new developments in gene editing. And we hear from Professor Ronald Conlon, who is in the Department of Genetics and Genome Sciences, as well as in general medicine. And he directs the Case Transgenic and Targeting Facility. Ron.
And now for something completely different. So the 2020 Nobel Prize in Chemistry was given for the development of a method of gene editing. And it was given to Jennifer Doudna of UC Berkeley and Emmanuel Charpentier of Max Planck in Berlin. This was a collaborative effort between the two. So the chemistry Nobel was awarded for the development, quote, for the development of a method of gene editing. What they specifically did is they identified, simplified, and reprogrammed a particular nuclease called the Cas9 nuclease that then made possible an improved method of gene editing. And this was a major advance. This research is really at the confluence of two different lines of work. One was work on an adaptive immune system present in bacteria and archaea, various microbes. And the way that gene editing and this Nobel Prize is usually presented is to highlight the evolution and our understanding of this adaptive immune system in bacteria. Because that's normally the way that this is approached, today I think I'm going to focus on the other stream of research, which is the stream of research that focused on gene editing. To be able to gene edit, we co-opt cellular DNA repair. And so to co-opt cellular DNA repair, we have to understand it. Key advances were made beginning in 1989 by James Haber of Brandeis University. He was studying the mechanism of DNA repair in brewer's yeast. So brewer's yeast has two sexes, A and alpha. And a yeast cell, a haploid yeast cell, can switch its sex. In fact, it switches its sex quite frequently at nearly every cell division. And so if you start off with a single yeast cell that is haploid, it can generate both sexes, and then the two sexes can mate with each other. To accomplish this switch in sexes, it takes a surprising route to do this. It cuts its own DNA, first of all, to switch the sexes. And this mechanism involves the insertion of a new DNA cassette into a player, where the player is a gene and a cassette is a component of that gene. And so a new cassette in the player generates a new sex. So again, yeast cuts its own DNA to switch sexes. This is a diagram of a yeast chromosome in a yeast whose sex is alpha. So the cassette player here, the gene that's determining the sex, has the alpha cassette in it, and it then results in the production of alpha factor, which is important for it determines the sex of the yeast. The first step in switching its sex, which it frequently does at cell division, is that the yeast generates its own DNA cutting enzyme, or endonuclease, which cuts its own DNA precisely within this gene that's making alpha factor. Then a cassette, the A donor from the same chromosome, is used at that cut in a process of homologous recombination to replace the alpha cassette with the A cassette. And now the sex has switched. We now have a yeast that is sex A because A factor is made in contrast to alpha. This process is reversed. The same cell then goes through the same process and it chooses the opposite donor. So if it's a sex A, it cuts its own DNA and it replaces the A cassette with alpha in the player. This process is known generally as homology-dependent repair. More precisely, for this particular process, it's called homologous recombination. The important thing is that DNA with similar sequence is used to replace the sequences around the cut. And so the DNA, you need two components here. You need similar sequence and you need a DNA cut. This kind of DNA repair was shown to be widely conserved. And so in 1994, Maria Jason of Sloan Kettering Institute showed that it's conserved in mammalian cells. And so she essentially recapitulated the same approach that James Haber used by generating a single site in the genome that could be cut by a DNA scissors or DNA endonuclease that she would supply, and then she would supply different templates. And she showed that mammalian cells could undergo the same process of homology-dependent repair. We co-opt the cell's ability to undergo this homology-dependent repair if we want to gene edit. Okay, so if we have a cell, and we're here representing a part of its genome with a gene in it as a linear thing with different colors representing different sequences, the first thing we need to do to recapitulate the process is we need to cut the DNA. So we need to cut it specifically in the gene we want to edit. Then we need to add DNA that's similar in sequence to the DNA we want to replace. And so this process is aided by cellular proteins, but in the end, what we're looking for is pairing between the bases of one strand of DNA with the bases on the strand of the other DNA. And so that's why homology, or the similarity in sequence, is important. If we didn't have homology, we wouldn't get pairing between these two. And so eventually, the one piece of DNA in the center is replaced by the other. The red is replaced by the green, and we have an edited gene. The two ends of DNA are joined together. So if it was known back in the 90s that if we had the ability to design a single cut in the genome, we could edit any site in the genome, because if we can get the DNA scissors in there, we can also supply DNA. So unlike the yeast, where the yeast is providing its own copies to switch into the player, the different cassettes that it's switching into the player, in this case, we want to supply the DNA at will. To give you an idea of the specificity that's required to cut once, the genome of humans is 3 billion base pairs for one set of chromosomes. So it's twice that for if you include both sets of chromosomes. And so at the top is the actual sequence, the A's, C's, T's, and G's of the actual sequence, and a mutation that's present and that causes cystic fibrosis. That's a small proportion of a gene that's over 250,000 bases long, all part of a chromosome that's 159 million bases long, and all part of a 3 billion base pair genome. And so we need tremendous specificity to be able to cut close to this mutation and add homologous DNA around that mutation so that we can replace it if we want to correct it. The other important thing that's often overlooked here is that to carry out this technology, we need to have the genome sequence. And so this shows then the announcement 20 years ago of the first draft of the human genome sequence. And since then, we have more complete, more precise, and more complete and more precise sequence of the human genome. And we now have genome sequences of many organisms, which then allows us to predict which sequences will be unique in the genome and unrelated to others, and therefore will be a unique site in the genome. And so we can target that. There were a number of different efforts to design these programmable DNA scissors or programmable DNA endonucleases. And so the first crack at it was taken in 2001 by Dana Carroll and Srinivasan Chandrasekaran, University of Utah and Johns Hopkins universities. And so they cobbled together a very clever scheme of using DNA recognition modules from one type of protein together with a DNA cutting enzyme with no specificity from another. And so these components come from bacteria and other organisms. And these were called zinc finger nucleases. So in a zinc finger nuclease, we have two different proteins. Here demonstrated as the left ZFN and the right ZFN. And they each have a nuclease domain here marked as FOC1, which by itself has no sequence specificity, but which will cut DNA. The sequence specificity is delivered by these components out here, which are repeating units, slightly different, of these zinc fingers. A zinc finger is a protein that can recognize a particular triplet of bases in the DNA sequence. So the blue module recognizes GTG, the red GCT, and so on. And so one can get a complete library, a fairly complete library of these zinc fingers and combine them in different combinations to almost any sequence. And then with a left zinc finger module and a right zinc finger nodule, and then the requirement for these two FOC1 domains to heterodimerize, we can get specific cutting here at a double strand break. So this was a very clever application of protein engineering to give us a programmable DNA nuclease. And these were reasonably successful in being able to achieve the goal of cutting once in the genome. A second crack at this was taken in 2010 by Dan Vetis and Adam Bogdanov. They generated new nucleases called talons, where the components are combined from various bacteria, and the components are put together in a scheme very similar to that with the zinc finger nucleases. So in this case, the protein modules recognize a single base instead of a triplet of bases. And so all we need are 4 different types of modules, and they can be combined in any sequence to have the specificity for any sequence in the genome. The advantage of these over the previous ones is that the zinc finger nucleases, the modules in that case, did not have true modularity. You could combine them with the hopes that they would work, but they wouldn't necessarily play well together. The talons were much more truly modular. If you combine them, they would work. The talons were only brought in 2010. By 2012, a new system was devised or discovered by Charpentier and Doudna. And this is the Cas9 and guide RNA from the CRISPR-Cas adaptive immune systems of bacteria. This system is quite different. There are 2 components to it. Here's our target DNA that we want to cut. We have a Cas9 protein and a guide RNA. In this instance, the specificity for the sequence specificity to the DNA target is provided by the Cas9 protein, which recognizes, first of all, this short motif here of 3 nucleotides, this AGG here, and then by base pairing of the guide RNA here to the target strand of the DNA. And then we get a cut right here at this point. The advantage of this is that it's very easy for us to make guide RNAs of any specificity, whereas it's much more difficult to engineer proteins. So making guide RNAs is scalable, making proteins is not so scalable. The improvements that with these different generations of programmable DNA scissors can be seen in the time that it took from their creation to the time of making gene-edited mice. So with zinc finger nucleases, it was 9 years before someone gene-edited mice with them, 3 years after the birth of talons before someone made gene-edited mice, and 1 year from the Cas9 discovery to gene-edited mice, which is pretty much the minimum time that if one can make gene-edited mice. The impact of CRISPR-Cas9 has been considerable. It has allowed gene editing in any organism if we have a genome sequence for it. And even in those model organisms in which we have fairly sophisticated genetics and the means of making mutations at will, it's accelerated. It's much faster. It's much easier. And so essentially in many standard genetic models, the mouse, Drosophila, the fruit fly, CRISPR-Cas has supplanted those. In addition, CRISPR-Cas9 has given rise to novel editing tools which are better suited for therapeutic use. And in fact, some CRISPR-Cas is already in late-stage clinical trials for genetic therapies. There's already at least one CRISPR-edited agricultural product that's on the market. And of course, ethical questions have been raised about the application to human biology. And that's it.
Well, thank you. Thank you very much. That's very fascinating story. So now we move on to our third speaker. The 2020 Nobel Prize in Physiology or Medicine was awarded to 3 scientists for their roles in the discovery of the hepatitis C virus, HCV, and how to get rid of it. And to hear about this, we turn to Professor Donald Anthony from the departments of medicine and pathology, who works on this virus himself. Donald.
Yeah, thank you. And I also have a perfectly timed career time span to appreciate the full impact of these 3 individuals. This is a picture of a infectious clone of hepatitis C infecting a liver hepatoma cell line, HUH 7.5. Charlie Rice has generously shared this cell line with many investigators around the world. We began to use this cell culture system in 2006, and I'll get into Charlie's contributions. But I think part of the reason that these individuals were chosen was in part their sharing nature of all of what they learned, so that there was a synergy between the work of each of them. When I attended medical school, my first year was in 1986, and at that time, hepatitis C was referred to as non-A, non-B hepatitis. We did not yet have the identity of the virus. We knew that it behaved in some ways like hepatitis A and hepatitis B. Hepatitis A, an RNA virus transmitted by fecal-oral method, was known over the 1970s, and Harvey Alter did contribute to the study of hepatitis A. Non-A, non-B hepatitis was understood to be something of some significance, but the full impact was not fully appreciated until work of Harvey Alter. Harvey was a dominant contributor to the understanding that non-A, non-B hepatitis was transmitted by blood transfusion. In fact, as I'll show in a few minutes, up to 5% of the blood transfusion pool in the country was contaminated with non-A, non-B hepatitis. My instructors in med school back in 1986 had the perspective that this was something that occurred, but the full impact of long-term consequences was not fully appreciated. And so the perspective was that the reason for a transfusion of blood was thought to outweigh the risk of the lifelong consequences of non-A, non-B hepatitis. Harvey's work was key in showing that this could be transmitted via blood. It could be transmitted to non-human primates, and the non-human primate system was a system that he used to be able to glean quite a bit of information. He developed a series of reagents of known infectious source from non-human primates and humans, all without having the identity of the virus or a test. It was all based upon tracing infection from one individual to another and looking at liver inflammation as a readout of transmissibility. In the late 1980s, Michael Houghton applied molecular biology techniques of that vintage to be able to develop a diagnostic test for hepatitis C. It was quite a tour de force. And what he did was to take a known infected sample and used random primers to make a DNA and RNA, a library of DNA and RNA, a cDNA library from a chimpanzee. And then he took serum from people known to be infected with hepatitis C, and he screened over 1 million different clones to identify a infectious nucleic acid that was used to develop our first generation test for hepatitis C, an ELISA-based test.
Sorry to interrupt, but I think the slide is stuck on the first page.
Oh boy. Should I stop sharing and go just to the...
You can share the full screen and we'll be able to see it.
Okay, is this easy? Can this be seen now?
No, you're not sharing yet.
Uh-oh. Share screen. I unshared and tried to reshare.
Yeah, you're back on now.
You're back on now.
Sorry about that. So was this slide at all seen?
No.
Oh boy. Okay. Well, so this is really getting back to my orientation in the 1980s, non-A, non-B hepatitis, not much known. And then through the 70s and 80s, Harvey Alter's contributions were, as I summarized, developing a whole series of reagents without ever having a diagnostic test. And then Mike Houghton's work of screening a phage expression system was the next advance. And the impact of this was in understanding the mode of transmission of hepatitis C, primarily bloodborne pathogen. And in 1992, the blood pool was cleaned up of hepatitis C. We began to understand that 85% of hemophiliacs had hepatitis C as a consequence of cryoprecipitate. We identified nosocomial outbreaks of hepatitis C, in particular in Egypt, and we began to learn additional epidemiology. The map shown here is a worldwide prevalence of hepatitis C, roughly 170 million in the early 2000s, and some particularly intense areas are shown. Egypt, where in the Nile River Basin, treatment of schistosomiasis with glassware that was reused resulted in over a 10% prevalence of hepatitis C. So Harvey and Michael, their work was completely transforming. And as I mentioned, the blood pool, 230,000 infections a year before 1990 in our country. And after cleaning the blood pool, less than 40,000 infections per year, currently less than 10,000 infections per year. predominantly through shared IV needles. So huge impact of these initial findings. We further began to understand that the consequences of hepatitis C are not really appreciated until 20 to 30 years after infection. In fact, some individuals resolve infection on their own, but most go on to chronicity, and a number of factors can accelerate the course of progression of disease. In some individuals, roughly 20% to 30%, cirrhosis or scarring of the liver develops, and after 20 to 30 years of infection, a fraction of individuals develop liver cancer or succumb with mortality. The process of cirrhosis shown here in the bottom right panel, a nodular texture to the liver due to collagenous deposition and scarring from chronic inflammation. The pathogenesis of hepatitis C was facilitated by these tests and the in vitro culture system that I'll talk about in just a minute. And we began to understand the host response to hepatitis C was a major driver of the damage. This has involved natural killer cells, CD8 T cells, intrahepatic Kupffer cells, plasmacytoid dendritic cells, and the concept of interferon playing a strong role in host defense of hepatitis C was quite appreciated early on. It was the therapy of choice in the mid to late 90s and early 2000s. We had a very wide variation in response to interferon-based therapy when we used it. Some individuals followed a trajectory of elimination of virus while on interferon over a year's time period and sustained virologic response, otherwise referred to as a cure. Some individuals, the viremia relapsed after cessation of the therapy. Other individuals had no response whatsoever. In fact, therapy was extremely unappealing. The side effects of interferon administration are a sensation of feeling like one has the flu. 30% of people had difficulty taking this therapy. Use of therapy, if one were to view this y-axis as a cure, the genotype 1 hepatitis C infection, we were only able to achieve therapeutic response in under half of the individuals that we treated. In fact, in more well-described populations, if somebody had cirrhosis of the liver, the chances of a response to interferon-based therapy were actually quite dismal. So Charlie Rice in the 80s was at WashU. He realized his study on the flaviviridae viruses was quite suitable for investigation of hepatitis C after the first clones of hepatitis C were sequenced. And Charlie applied molecular biology techniques and virology techniques to help develop the in vitro culture system for hepatitis C. He took a virus sequence that and was able to infect a chimpanzee, and he developed a full-length RNA clone of the virus. And then we, at around the same time, were able to identify a couple of clones obtained from humans that were infectious in a culture system. And the culture system that I showed at the beginning was our usage, where you can see the hepatitis C core protein stained immunofluorescence in the cytoplasm of cells infected, and adjacent or neighboring cells are infected. And this helped to understand that virus traveled from one cell to the next in a local fashion quite commonly. Charlie's work helped us to understand the membranous association of the virus complex. It's a 10,000 nucleotide, 3,000 amino acid plus strand RNA virus encoding a polyprotein product. There's a very long 5' untranslated region with an internal ribosomal entry site that facilitates translation. The protein product is post-translationally cleaved by a protease, and there are a number of virus packaged products that are necessary for replication. In fact, the RNA-dependent RNA polymerase is a target of the new direct-acting antiviral therapies that are a direct consequence of Charlie's culture system and work. The most commonly used direct-acting antivirals today are ledipasvir and sofosbuvir. There are tens of other direct-acting agents that are used. These agents, in combination with each other, resulted in a homogeneous response across all groups of individuals, 95% sustained virologic response or cure of hepatitis C. Comparing the time frame before 2015, when we did not have these direct-acting agents, to present, in our local VA population, we have 3,500 infected individuals. Today, we've treated over 85% of those individuals and have gotten rid of hepatitis C. The downstream consequence of hepatitis C, as I mentioned, is a lifelong infection in some, and over periods of 20 to 30 years, the full impact is usually appreciated. And so because of this time window, it's still anticipated the peak morbidity will be in 2030. But even at this point, we understand that removing hep C from the host, it reduces the chance of liver cancer by at least 50%. It improves the liver function within months, and the longer-term consequences are yet to be fully realized. So the transformation of a situation where we had no identity of the virus to a situation where we had reagents to detect and diagnose infection, and now ability to treat the virus. Of course, other efforts that still need to be tackled are vaccine and worldwide treatment of hepatitis C. But I think that the time period and rapidity with which these investigators contributed to these works is truly a testament to a collaborative effort. I'll stop there.
Well, thank you very much. That was very interesting indeed. So now we do have, thanks to everybody being so punctual, plenty of time for questions. In the chat function, I see that Walter has asked 2 questions, which were answered in the chat box, but I think maybe Walter would like to choose one of those questions and ask it himself now. I think you can unmute yourself.
Yes, thank you. So well, actually, Ronald Conlon already answered my question, but I was wondering if these techniques for cutting DNA had any impact on these new RNA-based vaccines for COVID that have been developed. And the answer seems to be no.
And not yet, but the CRISPR-Cas has transformed all of biology. There are a few people who are not doing it these days. And so in further research and perhaps in subsequent vaccine development, I'm sure that it will play a role.
Thank you.
Very good. I see Sophia has a question. Would you like to ask it? You're muted.
Sorry, my question was about the how the signals for the black hole at the center of our galaxy were taken. Like I know about the black hole picture where signals from telescopes all over the world are kind of combined to fill the sky. How was that done? Is it similar to that?
Yeah, it's similar to that. So by taking multiple telescopes and combining the light, you can get a resolution which is effectively that of a telescope that's as wide as the spacing between the telescopes. So they did that with this very large telescope array, combining all 4 telescopes into one, but with an advanced interferometer. So that's fundamentally the same technique they used to get that picture.
Thank you.
I don't see other questions in the chat box, so I'll try a trivial question for Dr. Conlon. If you have 2 sexes, why do you call them alpha and A?
Alan, you want to handle that one? I have no idea.
Okay.
Alan, you're muted. You're muted, Alan.
Yeah, Alan Tartikoff, we can't hear you. You're muted.
Here we go. I was just saying somebody must have studied their Greek. Other than that, I don't have a practical answer to the question, but it's interesting. There are closely related organisms that have more than 2 sexes. So there is deep and perplexing thinking with regard to these mating type interconversions.
I see. Very good. Let's see. Anybody else have a question they'd like to ask?
So Phil, can I just add that what Alan just said? So there are many of the Paramecia have multiple ones. So Paramecium bursaria, which actually my PhD work on, actually has equivalent of 8 different sexes, and they can work in combinations with each other. And so again, it's the same kind of process where you have a particular mating type that defines the sex.
I see. Well, opportunities for all sorts of ignorant, humorous remarks on that one. So, but are there some serious questions anywhere?
I have a sort of a career development question with regard to CRISPR-Cas. So to what extent were the laureates aware of the long-term possible utility of what they initiated long before?
So it was, so that sort of follows along the CRISPR-Cas lineage. It was suspected for a few years prior to their discovery that there was a nuclease involved, and certainly the work of other people suggested that there was a nuclease involved in the system. And it just, if you look at the sequence of the proteins, particularly Cas9, you can see that it has 2 nuclease domains. So it was really the understanding of the mechanism and demonstrating that it worked that was crucial to that, and then showing also that it could be reprogrammed, that it could cut sequences at will, that made it happen. In terms of, I suspect you're sort of alluding to the patent. Argue that certainly there was battles over who should get the patent, and so the patent office decided that applying what Doudna and Charpentier understood in an actual gene editing experiment was a non-obvious step, and therefore the patent for what they did went to the group at Broad.
Interesting. Well, Walter had a second question, which he had for Kurt.
Yeah, my question was about what was the technical breakthrough that allowed them to see these things, these star motions with such high precision?
Yeah, so it's really a combination of a lot of things. One is the wider availability of large telescopes, these 10-meter class telescopes, because you need to observe these things over 20 years. You need a lot of observing time on large telescopes. So the fact that those became widely available is one factor. The development of these advanced interferometers that enabled you to combine light from multiple telescopes. The development of the adaptive optics that you need and photographic techniques. So it's really a combination of a lot of things that made this possible.
I see a couple of hands.
I could add a little comment to that, which was pointed out by my former advisor from the Max Planck Institute in Stuttgart, and that is that Reinhard Genzel is the son of Ludwig Genzel, who was a director at one of the Max Planck Institutes, and he was a developer of some of the interferometry techniques in infrared region for, in condensed matter physics. And apparently, sometimes you can learn something from your father about interferometry.
Ah, interesting. Yeah.
I see Glenn Starkman has a question.
So coming back to an earlier question, if Cas9 is so ubiquitous in its use by biologists and going to be important for future vaccine development, why wasn't it important? Why hasn't it been important in the development of COVID vaccines?
I think that what was essential to the development of the COVID vaccines and their rapid development was the fact that the genome sequence of the COVID virus was made available very early. And then two groups jumped on that to make RNA directly from that sequence. So making RNA, as I said, with the guide RNAs is something that we can do very easily. And so they made mRNAs, packaged them in lipid nanoparticles, and essentially, I'm sure in a matter of days since they had the sequence, they were able to jump into trials in animals. It's really that rapid, and there was no need for gene editing beyond that. With a vaccine, what you want to do is you want to introduce the foreign protein or a bit of the virus that's non-pathogenic into humans so that the human will raise an immune response to it. That path is very well studied. We make a lot of vaccines. We're very good at it. Getting there through mRNAs was much quicker, and there was no need to gene edit along the way.
Thanks.
I see Yu Hao Sun has a hand up.
Hi, my question is, how do we measure the accuracy of gene editing result?
So we measure the accuracy of gene editing by sequencing the genome. One of the fortunate things about the CRISPR-Cas9 is that its off-target mutations that it generates are highly predictable. And they are predictable because they are related and closely related to, in sequence, to the on-targets. And so we can monitor a particular reagent in a therapeutic setting and ask, did we, in a preclinical therapeutic setting, did it have off-target mutations? And if it does, then we can refine the Cas9 and try again and see if we can get something more specific. The problem with the zinc fingers that is particularly a problem therapeutically, although it also has predictable off-target mutations, things related in sequence to the thing you want to get, it also has reliable off-targets at unrelated sequences for reasons we don't understand. And so that's much more dangerous. We cannot monitor those off-target effects by sequencing. Again, sequencing is something we do very well.
So is it like by checking the protein it transmits?
So having a sequenced genome, we have our target, and then we say, okay, given that sequence, we'll search like a word processor for a matching sequence and allowing a certain amount of mismatches in the rest of the genome. And so we can see if there are likely to be any closely related sequences in the genome. And then we can monitor specifically when we do an experiment to see if we've gotten any of those, if we've gotten any off-target effects. And so if there are no closely related sequences, you're probably good to go. If there are closely related sequences, the reagent may need to be modified a little bit to make it more specific.
So we can check the genome directly?
Yep.
Thanks.
Okay, we'll take one more question as part of the formal proceedings, and then we'll move more to an informal discussion if people are interested in pursuing that. Harsh Mathur has, in fact, a question.
Well, I don't know if it quite rises to the level of the last question for the colloquium, but I wanted to ask about, you mentioned that CRISPR was discovered by thinking about how bacteria defend themselves against viruses. So I was wondering if, well, first of all, is it surprising that there was a mechanism that bacteria use that was not already known? Or is it that there are many more to be discovered, and maybe this is a really fruitful area that we should all be, jumping into?
You should speak to the NIH. Yes, I think it's amazing that this adaptive immune system, this sophisticated system, was only recently discovered. The pressure from the NIH is always healthcare-related research rather than more basic research at all kingdoms of life. And so it's interesting that some of the key experiments that drove our understanding of CRISPR-Cas forward came from, first of all, European biologists who are not under such, pressure to make it all health related, and secondly, by commercial companies. So one of the key experiments published in the Science paper came from a yogurt company, microbiologists working at a yogurt company who had a contaminated yogurt culture, and they realized, well, commercial microbial cultures, they need to keep them pure. They can't have the viruses and stuff coming in there and spoiling their huge batch of yogurt. And so they hire a lot of microbial scientists. And so they did one of the key experiments to show that there's an adaptive immune system. And there's a lot more to be discovered there, because this is a highly diverse system. Only a few enzymes have been plucked from this highly diverse system of adaptive immune system. So there's a lot more to be learned from it still.
Can I add another piece in there, which also follows on? If we go back, the whole of biotechnology has actually stemmed from the restriction enzymes that were found in bacteria, where people weren't looking for some way of splicing DNA together. They were looking for, again, how did bacteria protect themselves from infection by viruses? So if this is sort of a 50-year span between the first restriction enzymes and then CRISPR-Cas, if you like, coming through, basically looking at this very similar response of microbes to infections in some form. And so I agree. I'm not sure that we understand the full suite of what things can do.
And there's other parallels to that too. The restriction modification system of bacteria, which was the basis of recombinant DNA, is essentially sort of an innate immune system, and then CRISPR-Cas is the adaptive immune system. And so they have similar functions in bacteria.
Well, thank you very much to our three speakers. This has been a most informative session. So well done. Thank you indeed. We'll stop the recording now, and anyone who would like to stay on and ask any more questions,